students, this is Miss Hudgens speaking. Right now I am in a meeting, so we are going to go ahead and continue notes. Um, so if you'll watch the screen here, remember we've been talking about living things. And yesterday we discussed the 6 characteristics of living things. So I want you to think right now. I'm going to give you about 20 seconds and I want you to see if you can remember the acronym and the 6 characteristics of living things. So kind of think to yourself in your head and see if you can remember what that is, okay? So starting now. Okay, we're going to go ahead and go on. If you'll remember, oops, that was loud. Um, it was Dr. Girl. So remember disposal of waste, reproduction, gas exchange, intake of nutrients, response to surroundings, and locomotion. And that's Dr. Girl. Okay, so now on to some other things that we had been talking about. In your textbook, they were discussing cellular organization. So you've already written down your vocabulary words, so you've identified what a cell is, but remember that it is the basic unit of structure and function for all living things. So yesterday when several of you were describing a car and saying it was not made of cells, you are absolutely correct. Living things do have cells in it. They're the basic unit. Um, if you'll notice here, on this slide, it talks about unicellular and multicellular. So some organisms are able to perform Dr. Girl and they only have a single cell. And so we would call those unicellular organisms. So bacteria, amoeba. If you look to the front of my board, I have visualizing protist poster. Most of those protists are single-celled organisms, okay, and they can perform Dr. Girl, and so they are living things. Now, for us, for many plants and many living things that you can actually see with the naked eye, which means you don't have to use a microscope, those types of organisms are multicellular. Okay, and we have trillions and trillions of cells in our bodies to make us the way we are so that we can be so large. Okay, so all of these perform Dr. Girl. Yesterday, I dropped a book and made a loud noise. Some of these noises that are coming through from my PowerPoint are maybe making you jump. That is a characteristic of living things, being able to respond to your surroundings. And so you'll notice here there's a picture of someone that is responding because the pan's hot. You'll also notice the here in the far corner the plant that is showing phototropism, and photo means light, and the tropism there is that they are moving towards the light. Okay, just like I said, all of my plants do before I kill them. Uh, the other one that we had talked about was the reproduction, and yesterday I told you normally we think about the sexual reproduction, the two organisms that will make a baby. They make an individual that has characteristics from both parents. Asexual reproduction means one parent and it's a clone. So you don't have to like write all about the stimulus and the response because those that was information that you have probably already written in your vocabulary. You might just write that that's involved in response to surroundings, but that reproduction, the asexual reproduction, we're making clones to reproduce. Okay? Now, the big study here, we've been talking about how scientists do experiments, and we, as scientists here in our classroom, have been doing experiments. But at one time, and this is talking about over 400 years ago, people thought that there'd be trash and garbage laying around, and they thought that living things could be reproduced or could develop from non-living things. So they would see trash out in the street and they would see a rat on it, and so they would figure that trash made rats. um, which in a way that's a good environment for them, so they would reproduce and live there, but they thought the actual trash made a rat. And so the same things would occur if they had rotting meat out. So they would see maggots on it, and so they thought that the meat produced the maggots. And so there are some very influential scientists at this time. San Francesco Redi did an experiment to help disprove this idea, and the idea was spontaneous generation. So they thought that living things, life could just spontaneously generate from non-living things. Okay, and so here Francesco Redi is does an experiment, and you probably have read about it. And let me show you this on the computer. Okay, and when I'm back with you, then we can go ahead and look at this again. We'll see if this is going to come up like it should. Okay, so here you can see Redi's experiment. And with this, we've got jars, we have meat, and we have lids. And he designed this experiment, so we'll choose these so we can do a test. And he designed this experiment to see if actually the rotting meat could produce living things, and he didn't think so in the first place, or he wouldn't have done the test. Okay, so here all of these things are the same, and then we're going to put a lid on the jar. Should I go ahead and put another lid on this jar? If I did, would I have the same results? Most likely. Okay, so my independent variable in this experiment, the one thing that I've changed in this experiment is my lid. Okay, that's the one thing I've changed. So I'm going to go ahead and run my experiment, and if you'll look, oh look, there's flies. And the fly on the jar with a lid can't get in there. And so over time it starts rotting and it's stinking, and both sides are rotting and stinking, but only the one that had flies land on it produced the maggots that then turned into flies. Okay? Now, another experiment. So that's Redi's experiment. And so he was it was a really awesome experiment there. Next, we had Pasteur. And Pasteur, we already talked about, I think, before, about pasteurizing milk. Okay, that's from Louis Pasteur also. He also wanted to check, because a lot of times they would have broth, and he wanted to check to see and show that if he had this type of a S-shaped flask, that there could be no bacteria that could get in. Okay, and so what he did is, because sometimes, when you have your broth, if you haven't cooked it, there's already bacteria in it, and so it can cause it to get cloudy. And so he decided that he was going to flame up this broth and boil out any bacteria that could be in it, and then it would cause it to not have anything in it, not spoil. Okay, so if you look over time, he's boiled the one on the right, and then the one on the left started to get growth in it. It wasn't boiled, so there was pre-existing bacteria. And then what he did, after that, and showed that no growth happened, he then broke the S-shaped flask off so that the broth was once again open to the air, and the bacteria in the air then infected the broth, and you could see growth. Okay, so that was his way to help show that there was living things that were in the air that could get into the broth and contaminate it. All right, so that was our spontaneous generation. Let's go back to that slide. So what we, ooh, that was loud, sorry. Stimulus and response. Um, so here, Pasteur did that a little after Francesco Redi, and this is the information about that. Okay, so controlled experiment, use boiled and unboiled broth in flasks with curved necks, and then it became cloudy. The one that wasn't boiled became cloudy. The one that was boiled was fine until he exposed it to the air again, and then it started to grow. All right. And if I'm going too fast when I'm talking about this, and some of you are needing extra time, you can always pause the video as you're going through. I just wanted to make sure that you were able to complete your notes. And I'm so sad I'm not there with you right now telling you all of this and seeing all your smiling faces because you're so excited about taking notes. Woohoo! Whew, that was loud again. Sorry about that. Now, the last thing that we're going to end on are the actual needs of living things. So we've talked about what it takes for something to be considered to be living, Dr. Girl, and now we're looking at what are things that living things have to have in order to stay alive. And so a lot of this is you should probably already know, you need water, you need to have food to eat, and organisms need living space, and homeostasis is also a big one. Being able to maintain your internal conditions. All right, and so a way that we ourselves, as we call ourselves endotherms. Endotherms means that we control our body temperature internally, and mammals um, need to keep themselves warm, whereas ectotherms like lizards and the reptiles, amphibians, they let the environment control them in their body temperature. So they can regulate their heat by laying out in the sun if they need to be warmer, and then whenever they're too hot, they can go in the shade or they go in the water. Okay, as endotherms, we regulate our homeostasis. Let's say we're hot, we've been running, we're hot, we sweat. When we are cold, we shiver, and that's our body's way of trying to regulate our body temperature and maintain homeostasis. All right? Um, so, that concludes our notes for today. Like I said, if you did not have enough time to write this down, they can always go back through and just show the little PowerPoint. I've got it attached to this. I just wanted to kind of talk to you about it as we went through. So if you have questions, please let me know. Hopefully I will be back in the room with you soon. Okay? Thanks. Have a good day if I don't